Hello friends, Ace Botany next episode. We are proceeding with the NCERT quick reference. And today's episode is Molecular Basis of Inheritance, sixth part, DNA replication first. And all discussion based on NCERT, reprint edition, December 2024, the latest edition. Initially, an introduction will be there, experimental proof and chemicals concerned with the DNA replication. Okay. Now, for convenience, I will share the screen so it will be easy for you. We can save time also. I think screen A. So we can proceed. Okay. So when we discuss this DNA replication first part, initially we'll be referring to a statement from NCERT given by Watson and Crick in 1953. That is a famous statement. It can be a question also. Okay, direct from NCERT. Now, three models has been there previously regarding DNA replication. One is conservative, other is dispersive, and third is semi-conservative. And most acceptable is semi-conservative. That means when the new double-stranded DNA form, one strand is new, other strand is old. And what is the experimental proof for it? That was done in 1958 in Escherichia coli using two chemicals. Basically, one is N14, other is N15. And remember, N15 is heavy isotope of nitrogen, but it is non-radioactive. And certification in a cesium chloride density gradient system. These all are direct questions by Matthew Messelson and Franklin Stahl. Meanwhile, a data that is ethidium bromide is commonly used to detect nucleic acids in molecular biology. In genetic engineering also, you may be studying about ethidium bromide. Taylor et al. In 1958, again, Taylor, another scientist, proved that the semi-conservative mode of replication by using radioactive time and to detect distribution of newly synthesized DNA in chromosome, performed on Visia faba root, commonly called faba B. And this experiment proved that DNA in chromosome also replicate semi-conservatively. So a very common question for theory entrance. Later, this type of experiments also done in higher organisms like plants and human cells. Direct NCERT, page number 8889, reprint edition, March 2024. I think idea clear. Now the experimental setup. So this is the experimental setup. We can find heavy nitrogen on both strands, allowed to replicate in N14 medium for 20 minutes. So DNA, that is two strands of DNA separate, one heavy DNA comes here, another heavy DNA comes here, into which new N14, because medium is N14, DNA comes. So in the first generation, we can find in a double-stranded DNA, one strand is N15, one strand is N14. Now again, it is allowed to replicate. So 20 more minutes, so total 40 minutes. Second generation, we can find this green will get another green, so completely N14. This red will be retained as red and a green will be added, so N14 and 15. Again, same with here also. And here it is both strands are N14. And this is another diagram. Here heavy DNA at the bottom of the test tube. Intermediate is at the middle area. And completely light DNA will be, or N40, N14 will be near to the lid. So that is indicating the distribution. And this is hybrid. So below N40, N14, we can find N15 and 14. And completely N15s will be at the bottom. I think idea clear. Now, based on this simple diagram, questions can be there. I put a question based on this diagram. If both strands of DNA N15 was allowed to replicate for two generation N14, that is why 1, 2, median, then ratio between N15 and 15, N15 and 14, N14 and 14 will be. So same question I put as a problem. Can be a good question for theory entrance. I think idea clear. Explanation we can find here. So we can proceed to next page. Now come some basic chemicals involved in DNA replication. This is not an exhaustive one, but this is based on or connected with our syllabus. These are the common chemicals. Enzymes I wrote there, helicases, their function, topoisomerase, DNA chirase, single-stranded DNA binding protein, ligases, primases, DNA polymerase in prokaryote, DNA polymerase 1, 2, 3. Polymerase 1 is called Kornberg enzyme, photograph I given there, North in 1959. Then DNA polymerase 1 also called Kornberg enzyme, etc. Then 1956 E. coli, first non-DNA polymerase main function is removal of RNA primer. So all details are given there. Then DNA polymerase 2 involved in DNA repair, and DNA polymerase 3, that is main polymerizing enzyme, fastest polymerization rate is around 2000 base pair per second. So can be a good question from this 1 or 2 questions we can anticipate. Theory entrance. DNA polymerase in eukaryote. 5 types are the eukaryote: alpha, beta, gamma, delta, epsilon. It's given there. Not in direct syllabus, but just mentioned there. Then deoxyribonucleoside triphosphate. 2 functions they can perform. One, it is substrate itself. Second, it can provide energy for polymerization. Common question for theory entrance. And 10th one is DNA-dependent DNA polymerase. Main enzyme, highly efficient, fast, and accurate. I think this data, better by heart it, there can be question for theory entrance. We can proceed to next page. Like ATP, the 2-terminal phosphate in a deoxynucleoside triphosphate are high energy phosphate. The whole prokaryotic DNA acts as a single replicating unit, or it is otherwise called replicon, while a eukaryotic DNA molecule has several replicons. Eukaryote and prokaryote, another difference. Analytical replication is a very expensive process, and we can find enzyme phosphorylase involved in activating 4 types of deoxyribonucleotides. And energy different forms, including ATP and GTP, will be utilized. Now, steps in replication, how they divide or duplicate, that we will be discussing in next episode. So if you're able to follow, please subscribe ACE Botany, like, share, comment on. And daily I'm putting 2 questions also in ACE Botany for NEET practice. So for time being, we can wind up. So take care. Bye. Thank you.